Welcome to Neoscience Hub. Today our editor Dr. Subramanian S. Iyer will be interacting with Mr. Ramalinga Prasad Kuppa, a GMP consultant based in Hyderabad. Mr. Ramalinga Prasad Kuppa has over 46 years of rich experience from pharmaceutical manufacturing organizations. He has worked with companies like ICI Pharmaceuticals, Apotex Paracom Inc., Ranbaxy Laboratories Limited, Sri Krishna Pharmaceuticals Limited, and Metrocom API Limited. His core areas of experience are in quality assurance, quality control, and training on all these aspects. Mr. Kuppa has expertise in understanding the exact interpretation and compliance requirement to the GMP regulatory challenges for all the ICH guidelines from ICH Q1 to Q11 and Udralex Volume 4, Part 1 and Part 2, etc. He guided the teams on concepts of process validations, cleaning validations, and support them for preparation of effective validation protocols and reports. Over to Dr. Subramanian S. Iyer.
Welcome to Neoscience Hub. Thank you, Mr. Kuppa, for being our guest today on this good show on ICH Q7, which is a document which actually explains about good manufacturing practice. Please tell our viewers how important is GMP for the pharmaceutical industry and what is the scope?
GMP is a good manufacturing practices. It is a requirement for all jobs of any activity, it should be there. But for pharmaceuticals, it is specifically required because we are dealing with the pharmaceuticals which are very important for the health of the persons using that. So it is very important that to follow the GMP rules fully as far as pharmaceutical manufacturing is concerned.
And going forward with what you said, if a manufacturer is not following Q7 GMP, does it mean his product is not safe?
Obviously it is not safe. The reason is when there is a requirement to comply to that, and if you don't comply to the requirements, there is every chance that the product can fail. And if a failed material or if a failed product is used by the ailing person, there is a possibility that it can have bad impact on the health of the person.
So it's a safety issue.
It is a safety issue and efficacy issue also.
Good. So I was going through the guideline. What I observed is there's a word called SHUG. Now SHUG in English has many connotations. What according to you is ICH interpretation?
Word should in ICH Q7 or in any other guideline of ICH, it is a recommendation that it should be complied with. That's a requirement.
Okay. So all over the globe, if anyone is following ICH Q7, they are supposed to comply with that.
Yes. You can have alternate procedures also, but it should be equivalent or better than what the guideline recommends.
So let's go one step now.
Yeah.
We are talking about API stacking material. That means for viewers, any manufacturing process, the first step which is called API stacking material. How important is this stacking material of API?
Starting material by definition is a major part of the API molecule. So if the manufacturing of key starting material, which is generally called in the industry, is also called a starting material. Some people will also call it as a regulatory starting material. Whatever way you call it, it is called a starting material. It should be a major portion of the main molecule of the API, and it should be freely available in the market, totally characterized with all impurities and all process requirements are totally set for that, and it is stable enough.
So does it mean that people who are manufacturing this stacking material are under the scope of GMP?
Yes, all the starting materials should be under the scope of GMP.
Very good.
Because KSM or starting material, most of the time it is almost N-1 category of the API.
So let's go about API per se. Is there a difference between expiry date and retest date of an API? If so, what is the basic interpretation, how people view?
Expiry date means beyond that period the drug does not have impact on its requirement. So hence it is not supposed to be used. Whereas retest period means after the expiry, after the retest period date, if it is tested once again and if it passes to specification, that can be used further.
So does it mean that a lab can keep on extending the expiry date?
Yes, the manufacturer can extend the shelf life to any extent depending upon the evaluation as per the guidelines. The guideline says that if there is no variation or significant variations in the results obtained from the accelerated study and long term study, the shelf life can be extended by some requirement. It is there. So as per that, as long as the product meets the specifications and as long as it is suitable, it can be extended.
Good. So talking of API now, as we all know, most of the APIs are outsourced. Now when a company changes the manufacturing process, how important is it for a buyer to know about this change and what are the implications of that?
When you are changing the source material from a different vendor, it is very important to see that the route of synthesis is similar to the one which was being used earlier. If the route of synthesis is not same, the impurity profile will be different, the solvents may also be different. So your specifications will not meet to those things, and you would have validated your process using the earlier steps. So it is necessary that when you are using a new source material, it has to be validated by taking 3 consecutive batches from the new vendor who has a different route of synthesis.
What does the ICH Q7 guideline say? Should it be calibrated before performing a preventive maintenance or after performing? How do you explain this? Because most of the chemists are confused.
The initial step of equipment is it should be qualified. It should be qualified for its design, for its installation, for its operation and for its performance. When once the performance is done, and it should be used for routine use. While using for routine purposes, it should be calibrated with a frequency as recommended by the vendor. And if it is not calibrated on time, that means the equipment is faulty. You cannot take the results which are obtained from that equipment as a proper results. That can be problematic results.
This calibration work, can it be outsourced or should it be done within the lab premises?
It can be done in-house if there is a facility to carry out the calibration. If it is not, you can have an outside calibration agency also. But it is very important that you should have some sort of an agreement with them on the terms and conditions, the frequency of carrying out, and he is also supposed to give you the procedure on how he is calibrating, and he should give the certificate of calibration also whenever he does.
Good sir, that was nicely explained. So going to the lab once again, I have noticed something very conspicuous. ICH Q7 does not talk about any lab notebook, any controls on the lab notebook. What is your take on that?
You are not right in that. The reason, if you see section 6.6 of ICH Q7, it clearly says that you have to maintain certain records for raw materials, intermediates, for testings. All those things are given in that. So there is a separate section in ICH Q7 that talks about requirements of records.
Okay, but what I was trying to say is the lab notebook has to be numbered, it cannot be duplicated, all those nuances are not captured.
See, it is a issue of data integrity. So all the registers that are used for recording purposes should be controlled. One step of controlling is pagination, so that even if you are removing some pages in between, you will be able to find out that some pages are missing. And also the register should be issued by a authority in the department, which is generally called a QA department.
Is R&D coming under the ambit of so-called GMP compliance? Are they supposed to follow the same thing which is being followed by manufacturing side in terms of quality systems and in terms of procedures and all?
As long as the product technology development is concerned, it is not really required to be a GMP compliant. But when you are planning to sell the product or market the product from the R&D stage itself, then all the GMP activities have to be done exactly as per Q7 guideline.
Even if I am not validating the method or something in R&D, you still expect compliance level to be high?
No, you have to validate the process. The process validation includes the cleaning validation, analytical method validation, revalidation, all those aspects have to be complete before the batch is being dispatched to the market.
Suppose I am not using an equipment for a long time. Does regulatory allow me to have a visual inspection, or one has to complement it by analysis?
It totally depends on how you protect your equipment. If the equipment is protected totally from any possible contamination from outside, a visual inspection may be okay. But in certain cases, there is a possibility of microbial growth also in that. In such cases, you have to prove that, or you have to establish that there are no microbial contamination, there is no bioburden. In that case, you can use that.
Even if it is not usually used, or if it's not frequently used, still you have to do a microbial contamination?
Yes, not for all, but you have to establish that your product is free from bioburden. It is very much required for sterile products. For non-sterile products, you have to at least establish that there is no bioburden in the product.
Very nice. What is the ICH talking? How do you evaluate the training of a chemist periodically?
As per the section 3 of ICH Q7, it is required to train the shop floor personnel on GMP and the shop floor requirements. The training evaluation can be done verbally if it is a simple procedure or simple SOP. If it is a technical analysis like a HPLC or GC, a coded sample may be given to the analyst. The results have to be reviewed against the original results, and if the results are okay, then the analyst is qualified. Otherwise, you may have to retrain the chemist to see that he will do the correct job. And even if there is a change in the procedure and if there is a change in the equipment, the chemist has to be trained.
Good, sir. I just want to touch upon some of your personal experiences. How do you evaluate a normal company, its compliance to ICH Q7 on a scale of 1 to 10? Where do you think companies stand?
It depends on the levels of compliance. If the company can comply to all the requirements from section 2 to section 19 of ICH Q7, that means the compliant levels are more. It can be given 9 or 10. And if none of these things are there, maybe you have to give 1 or 0.
So when you are exporting to the less developed countries, do you think companies should still comply with ICH Q7?
Yes, because sincerely, honestly, we are manufacturing pharmaceutical dosage forms. Dosage forms are used by human beings. So it is very important that you have to take care of health of the human beings. It's not the question of whether it is a regulated market or non-regulated market. GMP is the requirement all over for pharmaceuticals.
So let's talk about samples for clinical trials. At what level should GMP be there? It is not going for commercial, it's not going for patients outside, it's only for clinical trials. Where do you think GMP should be followed?
As long as it is not marketed and as long as there is no product going outside, the clinical studies can be need not be in the GMP level, but all the records should be available for any review.
So let's go to the most important topic, which is stability of a product. How important is stability of a molecule which is already in the market, and how does a pharmaceutical industry control the stability?
The stability studies is very much required for dosage forms as well as APIs, because the stability will indicate its suitability for drug product as well as the API. There is a guideline called Q1A-R2 that talks about how the stability studies are carried out. Generally, the stability studies are carried out as stress test. Number 1, stress test is normal temperature, beyond the normal temperature of 45 to 50 degrees, and that is to check the intricate stability of the product. If the product is stable, then there are 2 systems of doing the stability. One is an accelerated studies. In that system, the product is exposed at 40 degrees temperature and 75% RH for 6 months. So, during 6 months, every month the sample is taken and evaluated for the specifications, and if it passes, that means the accelerated study will be completed for after 6 months. The long term study will be done at 25 degrees and 60% RH. Of course, the 25 degrees temperature depending upon the different zones, the temperature will vary. So, in these cases what happens, for the first year they carry out the analysis for 3rd month, 6 months, 9 months and 12 months, and from the second year onwards they do 18 months and 24 months. From then onwards they carry out the test every once in a year. Like that you can continue any number of time. During this period, every time you have to take a sample from each station and compare the data with accelerated study, evaluate statistically, and then if the statistical requirements are met, there is a guideline in the guideline that is called Q1E, how you evaluate the stability of the product. In that, if there are no variations, if everything is okay and there is no significant impact on the specifications, you can give an available shelf life plus 12 months. For example, if you have a 12 months data, you can extend by another 12 months by giving 24 months as stability data for that. And if you have got only 6 months data, it can be extended by another 12 months. Like that, it can be extended by every 12 months. If there is a problem and if the data is amenable to statistics and if the data supports that there is no problem, then you can give X plus 6 months. So, there are conditions like that. So, you have to go through those conditions. So, the condition where there is no problem at all, you can give X plus 12 months. X means the available long term study data. So, X plus 12, X plus 6, X plus 3, like that for different variations as per the guideline. And if there are problems, you cannot extend the shelf life beyond that. So, this can be extended as long as the product is suitable and if it passes to specifications. All this information is provided in a flow chart in ICH Q1E guide. That is very important for evaluating the shelf life of the drug product or API. It is very important that the drug product should be within its expiry period before it is used. If you are using a drug product that is date expired, there are two problems can be there. One is its efficacy is reduced, or the impurities that are generated after the expiry can harm the health of the person.
So, you just spoke about impurities. What are the tests which is done to establish a shelf life?
The general test used for stability is, there is a requirement in the guideline that the test that is used should be stability indicating. That means when you are exposing the product at higher temperatures for longer periods, what kind of degradation impurities or what kind of impurities can be generated when exposed to the high temperature. So, generally the HPLC method is the one which is suitable for all these degradants. In these things also, the guideline prescribes that any impurity more than 0.10% should be reported and it should be identified and quantified. If there are impurities like genotoxic or carcinogenic impurities, all those things have to be controlled at much lower values. And for those things you may have to establish that whether or not your product is free from genotoxic, carcinogens and all. If there are carcinogens and genotoxic impurities in your process or in your product, you have to deal separately those impurities. It is not coming under routine impurities. There is yet another section which talks about other general impurities. They are all monitored at less than 0.1%, and as per there are several guidelines in the pharmacopoeia that describes more on that.
Sir, can you tell our viewers about, I heard that you have translated the entire ICH Q7 into the local Telugu language, which is a fantastic job. Can you please tell our viewers how you thought about this idea?
It was an idea that I got when I was going through some industry. many people felt that reading in English and trying to understand its intent and its content is difficult for them. Then I thought, let me translate that into easy language, a vernacular language, so that people can understand. So subsequently, I started translating ICH-Q7 into Telugu, that is our local vernacular language. Later on, I developed that guideline further by giving the requirements. That means the intent of the guideline is also provided. Based on the content, I described as a commentary how it should be understood and how it can be complied with. So it was almost 90 pages document, which is very useful particularly for people of Andhra Pradesh and Telangana, or whoever wants to know in Telugu. The very important point, the feedback which I got from that guide is, the people who read the Telugu guide, and if they read that English guide, they are able to interpret very easily, and they are able to understand the content and the intent fully well. So their compliance levels of GMP are very high now.
Fantastic. You have any plans to translate any other guideline also apart from ICH-Q7?
If required, yes, I want to translate more. One or two examples on ICH-Q9, that is the risk management system, systems and tools. I did some translation work. That is not full-fledged. I made only a small part of it, not much. But if required, I'll be able to do more.
That was excellent. Thank you so much for coming here and giving our viewers all the nuances of ICH-Q7 and how much work you have done. I appreciate you coming to Neuroscience Hub. Thank you so much.
Thank you so much. Thank you.